Pharmapal.
Multidrug resistance in cancer. Hey everyone, welcome back to our channel. Today, we're diving into a fascinating and critical topic in cancer research, overcoming multidrug resistance, or MDR, particularly focusing on a recent study that explores targeting a protein called P-glycoprotein. So, grab a snack, get comfy, and let's get started. First off, what exactly is multidrug resistance? Well, in simple terms, it's when cancer cells develop the ability to resist the effects of multiple drugs that are meant to kill them. This is a huge problem in cancer treatment because it limits the effectiveness of chemotherapy. One of the main players in this resistance is a protein known as P-glycoprotein, or PGP for short. Think of PGP as a bouncer at a club, deciding which substances can enter the cell and which ones get kicked out. Unfortunately, when it comes to cancer drugs, PGP often kicks them out, preventing the drugs from doing their job. The main goal of their research was to find ways to overcome this PGP-mediated resistance. They focused on developing new chemical compounds that could inhibit PGP, thereby allowing chemotherapy drugs to work more effectively. Now, let's talk about the approach they used. The researchers employed a chemical molecular-based strategy. This means they designed and tested various chemical compounds to see which ones could effectively block PGP's action. They conducted structure-activity relationship (SAR) studies, which is a fancy way of saying they looked at how the structure of these compounds affected their ability to inhibit PGP. They found several promising candidates that showed significant potential in reversing MDR. One of the standout findings was that some of these compounds not only inhibited PGP but also demonstrated good bioavailability and a proper half-life in animal models. This means they could remain in the body long enough to be effective without causing major side effects. In vivo studies. In their in vivo studies, which means studies conducted in living organisms, certain compounds were shown to reduce tumor size in models of leukemia significantly. This is a promising step towards developing effective treatments for patients battling drug-resistant cancers. First up, let's take a look at Figure 1. This figure shows the structure of P-glycoprotein, or PGP for short. PGP is a protein that sits in the cell membrane and acts like a pump, pushing out drugs from cancer cells. This is one of the main reasons why cancer cells become resistant to chemotherapy. You can see here that PGP has multiple transmembrane domains, which are like tunnels through the cell membrane. These domains help PGP bind and transport drugs out of the cell. The structure also shows the ATP binding sites, which provide the energy needed for PGP to function. Now, let's move on to Figure 2. This figure illustrates how researchers are targeting PGP to overcome multidrug resistance. They're using small molecules called inhibitors that can block PGP's activity, preventing it from pumping out the drugs. These inhibitors bind to the ATP binding sites on PGP, essentially turning off the pump. By doing this, the drugs stay inside the cancer cells longer, making the chemotherapy more effective. Now, let's take a closer look at Figure 3. This figure is split into two parts. Part A illustrates tariquidar analogs' general structure-activity relationship studies in PGP-mediated MDR. Here, we see how different chemical modifications can impact the effectiveness of these compounds in overcoming drug resistance. Part B showcases the chemical structures of selected tariquidar analogs that exhibit potent MDR reversal efficacy. This visual representation helps us understand how slight changes in molecular structure can lead to significant differences in therapeutic outcomes. For instance, the presence of specific functional groups can enhance the interaction with PGP, thereby improving the drug's ability to remain inside the cancer cell and exert its effects. Moving on to Table 1, we see a detailed summary of the reversal efficacy of various tariquidar analogs against PGP overexpressing cancer cells. Each row lists a compound along with its EC50 value, which indicates the concentration needed to achieve a 50% reduction in drug resistance. The duration of the reversal efficacy is also noted, providing insight into how long these compounds can maintain their effectiveness. For example, compound 183 shows an EC50 of 37.2 nm against the SW620/AD300 cell line, indicating strong efficacy. Meanwhile, the reversal fold, which tells us how much the drug's effectiveness is enhanced, varies significantly among the compounds listed. Next, let's explore Figure 4, which presents the chemical structures and SAR studies of tetrahydroisoquinoline-based PGP inhibitors. Part A outlines the general chemical structures, while Part B highlights selected compounds that effectively reverse MDR in cancer cells. These compounds are particularly interesting because they demonstrate how specific structural elements can lead to enhanced interaction with PGP, making them promising candidates for further development in cancer therapy. Starting with Figure 5, this figure shows the molecular docking of a small molecule inhibitor with PGP. Docking is like a virtual puzzle where we try to fit the inhibitor into the binding site of PGP. This helps researchers understand how well the inhibitor might work in blocking PGP's function. You can see here that the inhibitor fits snugly into the ATP binding site of PGP. This binding is crucial because it prevents PGP from using energy to pump out drugs from the cancer cells. Next up is Figure 6, which shows the results of a cell viability assay. This assay measures how many cancer cells survive after treatment with different concentrations of the inhibitor. The lower the cell viability, the more effective the inhibitor. In this graph, we see a clear dose-dependent decrease in cell viability. In simple terms, as the concentration of the inhibitor increases, the number of surviving cancer cells decreases. This indicates that the inhibitor is effective at killing cancer cells. Moving on to Figure 7, we have a bar chart showing the expression levels of PGP in treated and untreated cells. Expression levels refer to how much of the PGP protein is present in the cells. Here, we see that the treated cells have significantly lower PGP expression compared to the untreated cells. This means that the inhibitor not only blocks PGP's function but also reduces the amount of PGP protein in the cells. Figure 8 shows a Western blot analysis, which is another method used to measure protein levels. This figure complements the data from Figure 7, confirming that PGP expression is indeed lower in the treated cells. The darker the band, the higher the protein level. In the treated cells, the PGP bands are much lighter, indicating lower protein levels. Figure 9 presents the results of a drug accumulation assay. This assay measures how much of the chemotherapy drug accumulates inside the cancer cells. We see that in the presence of the inhibitor, there's a higher accumulation of the drug inside the cells. This means that the inhibitor effectively prevents PGP from pumping the drug out, allowing it to stay inside the cells longer. Finally, Figure 10 shows the results of a Xenograft mouse model study. In this study, human cancer cells are implanted in mice to evaluate the efficacy of the inhibitor in a living organism. We see that mice treated with the inhibitor have significantly smaller tumors compared to the untreated mice. This indicates that the inhibitor is effective in reducing tumor growth in a real-world scenario. To wrap up, the figures and tables in this paper provide invaluable insights into the ongoing battle against cancer drug resistance. By understanding potential inhibitors' molecular mechanisms and structures, researchers can pave the way for more effective treatments. Thank you for joining us today. If you found this video informative, don't forget to like, subscribe, and hit the notification bell for more updates on the latest in cancer research. Let's continue to explore the science that shapes our world.
Pharmapal.